most mind-blowing facts about our human brain is that it contains more neuronal networks than the billions of stars in the Milky Way galaxy. These neuronal networks are so important because they act as the electrical cables of both the brain and body to communicate. If these electrical cables fire out of control, it will cause what we call in medical terms a seizure. And when this uncontrolled firing happens again and again, it ultimately leads to the disease of epilepsy. Epilepsy is one of the most devastating brain diseases to live with that affects not only adults but children as well. And in Finland alone, at least 60,000 people have been affected by it. Currently, there is no cure, and with the treatment we have available today, 1/3 of the patients do not benefit. Just think about that. This problem is due to the limited disease testing models, such as animal models, that have partly failed in drug research. Therefore, it raises the need for us as researchers to develop not only improvement on the existing models, but more importantly, to shift towards human-specific epilepsy models. But in order to do this, how does one possibly model or mimic epilepsy outside the brain? My research aims to solve this by developing novel platforms. These new platforms consist of technologies such as microfluidic chips and microelectrode arrays. What are these? Microfluidic chips are small platforms that provide guided connections for the neurons so as to help us imitate how they're connected in the brain, like having a brain on a chip. Microelectrode arrays measure the neuron's electrical activity. So here, I use stem cells, which are unique cells of our body that can be programmed into any cell type, including neurons, which will eventually expand to neuronal networks. So by combining stem cell technology and these innovative platforms, I'm able to model epilepsy by using stem cells from patients that have epilepsy, make them into neurons, and grow them on the platforms. This advanced approach enables me to create unique profiles for each patient by comparing their electrical activity to those without and how they respond to different drugs and chemicals. My results show that these patients have very clear epilepsy-like behavior in their electrical activity. And furthermore, the relevance of this enables me to investigate certain biological questions associated to the disease. So now back to the bigger question. With this approach of modeling epilepsy outside the brain, I am hopeful and hope that you too are convinced that the impact of my research will not only improve our understanding, but more importantly provide relevant models to find effective treatment for those patients that have not benefited. Thank you.